Good morning, everyone. I think we're just a couple minutes past. Sorry, I'm Neil Neary. I'm the executive director of CAN. We're getting people are signing in. We're seeing some numbers. So I think, Crystal, we can maybe kick off with the opening remarks and such, and then I'm sure people will continue to join.
Okay, thank you, Neil, and hello, everybody. Welcome. I am excited to be here. I may know many of you from, especially the old hat I used to wear in working in neuroscience, but I am Crystal Phillips. I'm going to be your guide throughout all of these exciting programs for today. My main job is I am Vice President of Thin Air Labs Fund One. This is a venture capital fund where we look for all that great IP that you're developing, all of those great commercializable opportunities, partner up and commercialize these products so that we can create that impact at scale and get it into the hands of community members. So venture capital is my main hat, but the reason why I'm extra passionate about neuroscience is back in 2010, I founded the Branch Out Neurological Foundation. Many of you have been a recipient of the funds that we have been able to raise so that we can fund innovative neuroscience in universities across Canada. This was all inspired after my personal experience of being diagnosed with multiple sclerosis, navigating both conventional and unconventional healthcare systems, finding a gap and a need for more funding for some of the more alternative and high-tech innovations that all of you have been working so hard on. So that is my background, my passion for neuroscience, and I'm excited to be here and be wearing all of those hats today. Um, because of my experience with the Branch Out Foundation, I've also had the privilege of working with Campus Alberta Neuroscience and saw firsthand how collaborative they are, bringing together all three universities in Alberta, University of Lethbridge, University of Alberta, and of course, University of Calgary. They're doing incredible work, not only collaborating between those universities, but creating opportunities for neuroscientists to network amongst each other, collaborate with their research, and really just think bigger than say their laboratory space. So very excited to see Campus Alberta doing well, thriving, and continuing to see all this support and put these programs together. Now, before I dive in and talk about the schedule ahead of us, I would like to do a land acknowledgement. So in the spirit of reconciliation, we acknowledge that we live, work, and play on the traditional territories of the Blackfoot Confederacy, Siksika, Kainai, and Piikani, the Tsuut'ina and the Nakota Nations, Chiniki Bears Paw Goodstoney, and the Métis Government District 5 and 6, and all people who make their homes in the Treaty 7 region of Southern Alberta. Now, the program ahead is looking very exciting, and I'm hoping that it is not just you sitting back and listening with your screen off, but I encourage you to be a participant in today's program because we have incredible speakers, lots of inspiration. I know you're all curious minds, so I have no doubt that you will also have many questions. So that is the purpose of the Q&A chat here on the bottom of your screen. Feel free to use that, use the raise your hand, and I will try to facilitate and field and get as many of your questions answered as possible. Now's your chance because it is a compact group of experts and inspiring people to learn from. First, so I'll also just kind of run through what we have ahead before I introduce the executive director, Neil, and we will have, so we'll have presentations, we'll have some trainees speaking, and then of course we'll have those roundtables where you'll have the Q&A opportunities. So before we dive into our first speaker, Neil, I'd love to welcome you back onto the Zoom stage and speak a little bit more to Campus Alberta Neuroscience.
Thank you, Crystal. No, I appreciate you gave a great overview of CAN, and of course, we work really hard to combine the collective forces of the neuroscience community across Alberta and really strive to continue to have it be strong and promote Alberta as an excellent place for people to study neuroscience and launch neuroscience innovations. So thank you very much for agreeing to moderate today. We're very glad to have you. a few other people to thank. We want to thank Canadian Centre for Behavioral Neuroscience at the University of Lethbridge for sponsoring this event today. Really appreciate that. The Neurosciences Stroke Integration Network Program, so this is the PIN. Thank you to them for promoting, as well as Neurosciences Mental Health Institute at U of A and the Hotchkiss Brain Institute for being promotional partners. So I want to thank all of the presenters who agreed to come today. I agree we have a really exciting lineup. We're very excited to see Successes in Neuroscience come back. This was a program that was started a little while ago during the pandemic as an online forum, but it seemed to work out really well. So it seemed worth reviving. So, and then we're doing it today with one specific topic. I want to welcome the trainees. I'm very excited for them to get the opportunity to share what they're working on. I think that's going to be really interesting. And of course, the CAN team for doing the work to bring this together, but especially our very gifted summer student, Emma Patchett, who was with us for the summer and is hanging on a little bit to see this off. Emma is actually continuing the work of a previous summer student who actually started the first Successes in Neuroscience. So we were really glad to have Emma with us at CAN for the summer. And this was all of her doing. She did all the work and we're really appreciative. So yeah, and that's all I have to say. So I'll kick it back to you, Crystal, and you can go ahead and get things rolling.
Okay, thank you. That sounds great. And just a reminder, we have our first presentation coming up, and then it's going to be followed by a 5-minute Q&A. So at any point, get your questions in, and we'll have a very open dialogue going forward. And I'm very excited to introduce our next speaker, which is Dr. Martin Ferguson-Pell. I will let him introduce himself. Please don't be modest because it's impressive, just like Dr. Phillips, everything that you're up to. And he is then going to have a presentation on augmenting virtual care for rural communities, employing a clinic-to-clinic model, something I'm seeing more and more, very on trend. I'll let you take it away.
Thank you, Crystal. Can you hear me okay, Crystal? Great. And am I sharing or are you sharing the presentation?
You're sharing.
Oh, okay. Give me a sec while I do that then. And let me see if I can push that into presentation mode. I just had everything crash and I had to rejoin. There we go. Is that coming through okay?
Yeah, that's perfect. You're all.
Okay, great,
Oh,
fantastic.
thank you.
Well, my background is in biomedical engineering, but I've always been very close to the clinical interface in rehabilitation. And so I came to Alberta in 2007, actually to be Dean of the Faculty of Rehab Medicine. And so I've really had the opportunity to look at the field of rehabilitation, if you like, from a fairly broad perspective. And one of the things that really impressed me as, if you like, a city slicker coming from London, UK, is the incredible challenge that people who live in rural communities have in getting access to care in a timely manner. And so after I finished all of my admin responsibilities, I really wanted to look at how I could apply my background in biomedical engineering to help provide better access through virtual care. And we've adopted a very pragmatic approach to doing this. We wanted to keep it really simple in terms of the basic core technologies, and see how we can sort of build a platform that can then accommodate new technologies that can join onto that platform in order to provide a wide range of assessments. So let me see if I can advance my slides. Did that advance okay? Great. Okay, so in Alberta, which is a beautiful living lab in order to study this issue, around about 25% of our population lives in rural settings. And so when you start to do some analysis, so what we did was we said, imagine someone living 100 kilometers or more from an urban center, such as Calgary or Edmonton. What would the average cost for that Albertan, rural Albertan, be traveling into a single clinic visit round trip? Without counting in things like lost income, babysitting costs and so on, it comes out to be about $550. So this is using Revenue Canada expenses, models that enable us to estimate this cost. This is an out-of-pocket cost, not a cost that is picked up by the system. And so what are the consequences of this? Apart from the economic consequences, there is a strong tendency for people to defer getting timely care. And this results in, greater acuity of the problems that arise and poorer overall health outcomes for this group of people. When we look at the way we develop care models, one of the things that has really struck me is that those people, and I've done it, who sit around a table and come up with a care pathway are predominantly urban. So we come up with models of care, care models, if you like, that determine the various stages of the way we deliver care, that are very much centered around the way we live in an urban setting. But for people who live in rural Alberta, especially farmers and ranchers, they're dealing with the consequences of the fact they're working with nature. So they have harvest, and they have calving, and they have planting, and so on. And these are all things that are a little bit out of their control, yet they have a profound effect on their ability to manage their economy. And so what this results in, and there's a lot of evidence to show this, is significant anxiety and mental stress for people who are in rural communities who are farmers and ranchers. And so this is an example, but there are many other examples, including physical examples, of where we're not paying enough attention to how we deliver care that is supportive of the ways of life that people who live in rural communities have to manage. And so my lab is an interdisciplinary lab at the University of Alberta. It's basically a sandbox where we develop a whole range of different technologies, but we have a strong commitment to implementation. So we have about 25 running projects now in our lab. Many of them are centered around virtual care. Why is it important for us to provide virtual care? Obviously, it's the value associated with equity in terms of access, but also decreasing those healthcare costs that relate to some of the higher levels of acuity that we see in rural populations because of delays in accessing care. And we know, because we've seen this and we've heard this from people we've spoken to, that people die on the road going to urban-centric patient clinics in the winter because of bad winter driving conditions. So it's not just an inconvenience, but it's a hazard. And so there are real imperatives for us to look at ways of reducing that risk, which of course we can't do for everything, but maybe we can do it to some degree. So our aim is to improve access, to make it a financially sustainable program so that it's not something that we can just do as a project, but something that we can implement within the system. And then look at making sure that the way we deliver virtual care is as good or better than it would be if that person actually attended in person. So that we use a clinic-to-clinic model versus a clinic-to-home model because we want to be able to do complex assessments. So if we do it clinic to home, we're tied to the home environment, the lack of support for that person in terms of their safety, and also the various IT issues that can occur in a home environment. So the clinic-to-clinic model is one where we would have an urban specialist connecting to a generalist in a rural health center or a rural primary care center, where the technology can be put in order to support the assessment, and the connection is made synchronously between the expert clinician and the generalist who is supporting that patient in the rural community. The benefits also to the rural community is this begins to build networks to reduce professional isolation and build communities of practice. So it means that that person who may be not supported by a large number of colleagues around them to help them with complex cases now has a place to go, has relationships that they've built in order to be able to get support and feel less isolated in the way they deliver their practice. So we've been doing this now for about 6 years. So we preceded COVID. We didn't climb on the bandwagon. We were actually struggling to get people to believe in the importance of virtual care. COVID came along, and that certainly changed things a lot. But the post-COVID syndrome now is that the system is so stressed that the ability to actually accommodate innovation is quite limited. And so there's a tendency to revert back to the mean and go back to the way things were done before COVID. So this is an ongoing struggle to achieve implementation within the system. But we do have 15 sites around Alberta that we put the technology into in collaboration with Alberta Health Services. So what our platform looks like is basically a cart, as you can see in the picture, with a mobile robot, a double robot, which is manipulated by the urban-based expert clinician to move around the room, be able to look at the patient as if they were in the room. And the other benefit of this is it creates what's called presence, which means that there's an actual sense of that person who is coming into that rural community actually being there. And that's what our patients report, that it gives them a sense of confidence in the relationship with their expert clinician who's advising them. Also, it improves the audibility of the communications because the person is able to see the expert clinician close up, be able to pick up non-verbal cues, which helps with hearing, and also it gets the speaker system closer to where the patient is, so that for seniors who are hard of hearing, this improves their experience. One of the other technologies that we use is markerless motion capture, where we are actually able to make measurements of things like range of motion, balance, gait, other parameters as well, that transmit quantitative, objective data from the rural community to the expert clinician, and enables them to build that information into the assessment. We're working with the Rapid Access Clinic program that Alberta Health Services are rolling out right now in order to improve access to MSK conditions, for example. And I won't go into the details of this other than to say that this is going to have a significant impact in shortening wait times for their first assessment, and also reducing the need for people to have to travel in for a relatively simple assessment to determine if they're a candidate for surgery for their MSK condition, like OA, hip and knee, maybe problems with their shoulder and so on. We can also do virtual complex wheelchair seating assessments with this technology, and this is a very important requirement that Alberta Health Services has identified. We're also providing to Supported Living Centre, actually 4 of them in rural Alberta, pelvic floor assessments and exercise programs to reduce the challenges of stress incontinence. We've also built a technology to enable us to start to do assessments of people who have had a stroke and are experiencing upper extremity spasticity. And this enables us to perform what's called the Ashworth scale assessment, which looks at changes in spasticity that occur over time, but which can be treated with Botox treatment. And so what we're doing right now is to assess patients remotely to determine the right timing for them to come in and get their Botox injections. And the long-term goal is to be able to provide guided injections remotely, so that it's not necessary to travel in for the injections as well. The first step in this is to produce the means to be able to assess the level of spasticity and the readiness of the patient for that treatment. There's a real challenge in implementation when you're working at it from an academic perspective, and that is because of the way we're funded. The perception is that we're here today and gone tomorrow. We'll do our project, we'll show it works, everyone gets excited, the funding runs out, we've gone. And that's a real challenge in terms of how we can build confidence in what we do so that we can sustain these programs and over time get them implemented. The typical grant is far too short to get something absolutely embedded within the system. That's a 5 to 10 year process, and we're usually funded 1 to 3. So this is one of the challenges we face. We're trying to overcome that. Other things we've encountered are things like compact clinical areas where it's very tight in terms of space. Another one, funnily enough, at the very end is bright, shiny floors. They do a great job cleaning the floors, but the trouble is that the robot gets too much of a reflection back. So these are the real world challenges you have to overcome if you're trying to get something to work and something to stick. And so one of the things that we've been doing is, in addition to working with AHS, is working with primary care. And primary care is very keen to work on these challenges and actually to host these assessments. So we're working with primary care centers in Airdrie, Vulcan and Peace River right now to get things off the ground so that we can look at the alternative primary care model versus the AHS health center model. This is our team, group of students, trainees, and staff members that all pitch in to make this work. If you'd like to get more information from us or make contact with us, please contact Emily Armstrong or myself. We're readily available, and these are all the people that have helped to make it happen. Thanks, Crystal.
Oh, are you ever punctual? Thank you, Dr. Martin. This was incredible. I think the theme that I'm picking out in your presentation and the work that you do is, and it's reflective on this screen right now, is collaboration. Not just as a word, but true collaborations. I saw various startups, Kinetisense, that you're working with. You're working with the government, you're working with academia, with other scientists and researchers. I'm almost, my head spinning from all of the impact that that is creating in various ways. And so an obvious question to mind is how do you collaborate with everybody? Do you have any advice for other people who want to adopt a more collaborative mindset?
Yeah, I would say, and this is sometimes difficult for us to do as academics, in that I've actually made sure in our funding that we fund the right level of project management support for our projects, so that it's not all falling on me to be the person that's making all of these connections. So at the moment, our program has 4 project managers who help to build these relationships and sustain them. And so it's important when sort of costing out projects and funding projects that we build that capability in. All of our project managers have, significant experience in academic training, so they're able to look at things from an academic perspective. So they're not administrative from the standpoint of, scheduling and minute taking. They're actually getting directly involved in steering the projects. And so I think that's the key for us in terms of being able to manage this volume of connections.
So it sounds like you really do play people's strengths, including your own strengths, and you have this relay team mindset as opposed to thinking when it comes to something as complex as the healthcare system and population health, that it isn't one organization or one person who's going to make all of the necessary changes.
Not at all. I mean, regardless of whether you're working within the AHS system or you're working with other entities like the primary care centers and so on, a lot of effort, a huge amount of effort goes into relationship building and navigation, finding the right people to become champions. And of course, one of the things that makes that challenging is systems are not stable, right? So people keep changing and roles keep changing and so on. And so this is an ongoing, full-on effort in terms of trying to make those relationships stick.
I think that's a really good advice and mindset to adapt. So, and I think adapt is the word there. Things are always changing. You're always looking to collaborate, but you're ready. And in that athletic stance, which means that you're always ready for those changes, and you're in a good, strong position to be able to follow those changes.
Yeah, I think, yeah, looking at it from an athletic perspective is a really good way. You got to stay fit.
And flexible and strong,
Yes.
and resilient. Yeah, all the
And,
buzzwords.
and committed and focused on the prize, right? And
Yeah,
for us, the
exactly.
prize is that rural Albertan that is struggling to get care. I mean, we have certain values that are central to our program. Access is number one, and equity is number two. So I think that we try to live those every single day in our lab, because those are the things that kind of generate the energy.
I love that. Thank you so much. Now, we have another opportunity to hear you speak. Dr. Phillips will be, he just had to step away to address something, but we will have some more Q&A time after the next presentation. So if you stay online, would love to hear more, and alongside 2 other great speakers. So thanks again. Great presentation, and we will see you soon.
Thanks, Crystal. I'll hold on.
Okay. All right. So this is an inspiring start to the morning. I don't know about all of you, but definitely is for me. I wish I could see all your smiling faces. I'm sure you're just beaming. This is all your world. Now, before our roundtable discussion, we are going to invite Jordan Dudley, who is going to be our trainee spotlight, our first trainee spotlight, and he has 10 minutes to present his research before we all, the 3 of them, if Dr. Phillips is back, and then Dr. Ferguson, Pell, and you, Dr. Dudley, or sorry, maybe not doctor yet, will be all in our roundtable discussion. So I'll let you take it away for the next 10 minutes before we regroup.
Thank you very much, Crystal. All right, hello everyone. My name is Jordan Dudley, and I'm an undergraduate student at the University of Lethbridge. I'll just share my screen here. I work in the Wishaw and Mohadrani labs at the University of Lethbridge, and it is a pleasure to be able to present to you my research today. My project is goal tracking in an experimental autoimmune encephalomyelitis mouse as an early fatigue symptom of multiple sclerosis. Multiple sclerosis, or MS, is an autoimmune disease that causes physical and mental deterioration through damage to the nervous system. A common symptom of MS is fatigue. This disease does not yet have a cure, and thus we look to study it further in hopes of finding better treatment options. In order to better understand MS, we turn to a mouse model known as experimental autoimmune encephalomyelitis. Normally, the progression of this disease is tracked through a physical mouse hanging task that produces clinical scores on the mouse's physical deterioration. However, using this technique, the onset of symptoms is spontaneous and followed by a very rapid decline, leading to a very short time frame of intervention and research. We therefore ask, if symptomology can be detected earlier, could treatment options be evaluated earlier as well? In hopes of gaining insight into the progression of EAE, we had 21 female C57BL6 mice undergo a string pulling task. In this task, the animal is put in a box wherein it has to pull down a string hung above them in order to receive the food reward tied to the other end of the string. We had 6 control mice who received a saline injection and 14 treatment mice who received a Hook Labs EAE immunization. All mice were pre-trained in the string pulling task and had similar performance levels going into the experiment. The mice performed string pulling every day for up to 34 days. If a mouse received a score higher than 7 out of 10 on the mouse tail hanging task, they were removed from the experiment for ethics purposes. Of the EAE mice, half of them were dubbed asymptomatic through the mouse tail hanging task, and half were removed before the end of the experiment, dubbed symptomatic. Let that load. Here are 2 videos, one taken from a control mouse performing the string pulling task, and one taken from a symptomatic EAE mouse. Looking at these videos, we made some observations. The control mouse on the left completes the task in 1 run, from start to finish. Start it again. The mouse does not take any pauses, standing efficiently and pulling the food down. The EAE mouse on the right, on the other hand, takes pauses and sits down on the floor to smell the string or pauses in its movements while standing to smell the string. It appears as though, rather than habituate to the task, like the control animal does, the EAE mouse is falsely tracking the string. Searching for an explanation for this behavior, we found a behavior known as sign tracking. The average mouse performs goal tracking, wherein they will habituate to a task and recognize it needs to complete the task to receive the reward. A sign tracking mouse, on the other hand, will look for the reward, or in this case, the trigger for the reward, without having completed the task. This behavior can be attributed to fatigue. Therefore, if we measure the number of sign tracking events and see a pattern, we can see if fatigue measured through sign tracking events can be considered a symptom of EAE. During the string pulling task, we thus counted each instance of sign tracking, defined as an instance in which the mouse dissociates from the string it was pulling, either turning its attention to and sniffing the already fallen string, or pausing and sniffing the string in hand. You can see on the slide here, the 2 on the bottom right are examples of sign tracking, while the 1 on the left is goal tracking. We put these numbers into a cumulative graph, which, as you can see, showed some very distinct differences between the control mice in purple and the treatment mice in teal and red. What more, we found that the supposedly asymptomatic mice in teal with clinical scores reaching no higher than 1 have a statistically significant difference from the control mice and the symptomatic mice, suggesting they weren't actually asymptomatic, but that the progression of the disease in these mice simply wasn't identifiable through the mouse tail-hanging task. Ultimately, the control group in purple had a consistently low number of sign tracking events. The EAE quote-unquote asymptomatic mice were in the middle, and the EAE symptomatic mice had the highest number of sign tracking events. Running ANOVA tests on day 14, denoted by the green line, we found each group is statistically distinct from each other group, as confirmed by a significant p-value. What does this mean? EAE-treated animals displayed more sign tracking events than the control group. These sign tracking events are puzzling, but a potential explanation is that in string pulling, it takes a lot of effort to find the goal, and thus sign tracking may be attributed to fatigue, which is a pronounced symptom of human MS. Fatigue may, therefore, be a valid method of measuring the progression of MS, and sign tracking as a method through which to measure fatigue may allow us to measure MS progression. This could open up new doors in MS research if this phenomenon translates to a human model, which would require future experiments to confirm. Perhaps sign tracking would allow us to identify MS earlier in human patients, giving us more time to analyze its pathology before physical symptoms appear, and allowing us more time to treat MS, and potentially giving us the opportunity to perform earlier research and thus gain insight into the cognitive implications of MS. Thank you.
Well done, JC. Thank you so much for your presentation. For what it's worth, from an anecdotal perspective, I can totally relate to that AEE mouse sometimes.
That's fair.
I hope that this leads to the type of research that you're hoping for. And if you stay on the line, we might have some other questions from the audience. And this is a reminder to the audience, please put your questions in the Q&A here. Now's the time, because I'm going to call Dr. Phillips and Dr. Martin Ferguson-Pell back up for a roundtable discussion. So if you're there, please put your screen on, and we can get started. Dr. Phillips might need a couple more minutes here if he's able to make it. If not, it's the three of us. So I am going to start with a question that's just a little bit personal, and it's a question that really fascinates me, and it's the why you do what you do. So I'm going to have you both answer the question of, how did you become a neuroscientist? Why are you a neuroscientist? What gets you up in the morning, gets you excited about this field? And after I have both of you answer this question, I'm actually going to have you answer or ask the other person a question. So Dr. Ferguson-Pell, if you don't mind asking JC Dudley a question, and vice versa. And that is how we are going to start this roundtable before some audience questions. So first up, why do you do what you do? How did you become a neuroscientist? And Dr. Ferguson, I'll have you start.
Okay. Well, I think, but it's an evolution that, occurs during your career. So I don't think it's a sudden, in my case anyway, it wasn't a sudden decision. It was opportunities that I had that brought me into the field of spinal cord injury related research. And I think what I enjoy about it most, and what really attracted me to this field, is complexity. And I don't mean complexity just in terms of really difficult problems that we're trying to solve in terms of one single discrete problem, but it's really the complexity of the whole circumstance, if you like, associated with the clinical condition that we're working with. So one of the things that I'm fascinated by is the complexity of the life of a person, for example, with spinal cord injury after they're discharged from acute rehab. So there's complexity in acute rehab for sure, but when that person starts living their life outside of acute rehab, there is a huge complexity that most of us don't have to work with. And working with that and trying to unpuzzle that and help people overcome those barriers and those challenges, I think is one of the things that I find very rewarding. And really calls upon a lot of creativity. And when it's combined with technology and how technology can help us with some of that puzzle solving, I think that's what gives me the greatest joy. So for me, it's the big picture and how to apply technology to help solve some of those big picture problems that people are encountering in the neuroscience area.
You can definitely see that passion come through with the type of work that you do. So thank you. How about you, JC?
Hi. I'm just, I guess, starting. I'm in my undergrad, my 4th year at the moment. So I've done quite a bit of neuroscience research, relatively, having started in my 1st year. But what I'm continually interested in is, as I learn more and more about neuroscience and its implications, and as I do more research going into the intricacies of how neurodegenerative diseases take place, is also very similar to what Dr. Ferguson was saying there, in learning how it impacts people's lives and hoping that at some point this research can be used and implemented in ways that can help people to cope with how the disease is progressing, to help them live, I suppose. That's a very broad way of saying it, but I feel as though my definition of what I like to do will probably expand or get a little more precise as my career progresses. Thank you.
I'm sure many people can relate to that. Okay, Dr. Ferguson, do you have any questions for JC?
Yeah, I wonder about this one, and it's a difficult question, so please forgive me asking a difficult question. But in the work that you're doing in MS, I guess there are sort of two channels of work, right? One of them is how do we help people to overcome some of the limitations in their life that MS brings to them through the use of accommodating solutions, if you like, versus going for the cure. So from your standpoint, which direction do you intend to take and why?
That is definitely a difficult question. I think as of yet, what opportunities I've had in the lab had led me to learning more about the pathology of MS and therefore searching for ways to intervene earlier on, or in this case, find symptoms earlier. But from that starting point, it could arch out into both directions. I think finding the cure is definitely, I suppose the cure is a broad term, but finding that is always a very desirable goal, but at the same time, we don't know when we're going to get there, and that could be a very long path. So finding intermittent ways to make lives of people with MS better is very important as well. And therefore, I suppose it'll depend where I get accepted for graduate studies and what opportunities are presented to me. Both are very interesting in my opinion, and I'd love to help however I can in either direction. Thank you for the question.
Great answer. Now, JC, do you have any questions for Dr. Ferguson?
Trying to think of any insightful questions, but I suppose with what you're working on currently, what is kind of your next goals or upcoming goals that you'd like to achieve or accomplish?
Great question.
Yeah, that's really interesting. I think that there are quite a few. So some of those are to do with implementation, and that's getting it to stick. At the moment, I would say things are tacky rather than really sticking. And so one of the goals is to try and overcome whatever barriers there are to really change the way we look at the delivery of virtual care. And I think coupled with that, I see really interesting opportunities to transform the way we provide rehabilitation assessments and services. One of the difficulties we have in rehab, and obviously neuroscience is a component, a strong component within that, is that so much of what we do is observational. So much of the synthesis is done in the head of the clinician, and the available evidence to support some of the things that we do is fairly flimsy. And what I can see through virtual care work that we're doing is the need to transmit data that we've never had to transmit before. And so we have to make measurements that we previously judged. So an example of that would be, someone has got a sore shoulder. So let's see how sore it is by measuring how strong they are when you push down on their arms, right? It's called the Job's test. And you can judge that subjectively, but you could measure it in newtons. We never measure it in newtons clinically. We just say, oh, it's about 20% stronger on the unaffected side versus the affected side. We never know what that force is. And we never know really what the force-time relationship is as we push down. And that might give us some useful information about, something that suddenly gives at some point, at some angle, or some velocity. And so I believe that hidden within practice is so much information that we could get access to that would be giving us much greater sensitivity to changes that occur within someone, and also more specificity in looking at, what the fingerprint might be for one type of pathology versus another type of pathology. And so virtual care is forcing us to some degree to make measurements we've not made before. So what I would love to see is the acceptance of those measurements, and to get that, we need normative data, which means we need to get this going at scale in order to get enough normative data. And then I think the other part of it is how we can feed that information back to clinicians so it's meaningful to them. Clinicians haven't got the time to just collect information or data for data's sake. They really want data that is going to add value to their assessment. So I think the challenge is collecting it, synthesizing it, and feeding it back in a meaningful way. And maybe virtual care is one of the ways we'll start to change the way we deliver rehabilitation services and build greater confidence and trust in the assessments that are made that are currently based on judgment rather than measurement.
The CAN team has a follow-up question to that, and I think it's a big one, and one that I ask all the time when I'm wearing my VC hat. And it actually reminds me of Dr. Eric Topol's book, Deep Medicine, and it's how to put AI in health and how it puts the human back in healthcare. So everything you're talking about, you must be thinking about, how do I scale this? How do I create more efficiency and effectiveness? You alluded to it in your previous answer, but I'd be curious about, if you were to layer in AI, which you may already be doing, what changes do you see coming?
So yeah, I think that's the exciting opportunity, right? So to make AI work, at a level that would be clinically meaningful means we have to collect a lot of data. So we have to scale up. And that means that everybody needs to get on board. So it's not just, a bunch of boffins in a lab doing some work. It means that the system as a whole needs to recognize the opportunity and create capacity to do it, which is not easy to do in the current clinical climate. But assuming that we can start to get to scale and start collecting larger volumes of data, the opportunity I think AI has is for predictive analytics. And so an example of that would be, let's say we're collecting information on a particular way a person is functioning. So let's say it's someone who's had a spinal cord injury, they've been discharged, and we're collecting information about the level of tone associated with their spasticity. And we've got a means of measuring that remotely, so they don't have to keep coming in for a visit, but we can just get that information. What we could do is watch that trajectory and see how that tone is changing, whether it's getting better, whether it's worse, or whether it's plateauing, whether the plateau level is considered to be functionally acceptable or unacceptable. And on the basis of those trajectories, be able to predict whether or not we need to change the treatment for that person, how much we think that that is going to affect their ability to function. Now, the wait list right now for someone who's been discharged from somewhere like the Glenrose with spasticity associated with a spinal cord injury is about 8 to 9 months. So if that spasticity starts to build up and become a problem, the place that will be able to provide them with more immediate support is probably primary care or the emergency room if they have access. And they do not have a great deal of in-depth experience in working with relatively rare conditions like spinal cord injury. Most primary care docs may see 1 or 2 spinal cord injured persons in their whole career. So where AI could help with this would be to say, we can't get this person back to the Glenrose at the moment. The wait time is too long and the volume is too high. What we can do, though, is provide trusted AI to enable us to inform you as to how best to manage this spasticity for this person, and that this process or this guidance is supported by the authority, if you like, that the experts who provide these services can endorse. So a great example of this is autonomic dysreflexia. So imagine you're a person with a spinal cord injury and you're experiencing the onset of autonomic dysreflexia. If you need to get emergency help for that, you go to the emergency room. Likelihood is the person you're going to encounter in the emergency room has no idea what autonomic dysreflexia is, and also how to stop it. And it's not treated pharmacologically. It's treated by removing the thing that triggered the autonomic dysreflexia in the first place. And so that's the vulnerability that people have that I think that AI could help us to bridge. It's not as ideal as actually seeing a knowledgeable expert clinician, but given how scarce that expertise is, maybe AI can help to fill that gap, or maybe other AI supported by virtual care technologies could support the solution to that. So that would be a very long answer to an interesting question.
Well, I hope you're the author of the next AI in medicine book. Comes out. Jordan, there's a question from the audience to you. What are the challenges that you have found in your research about MS?
Certainly. Thank you very much for the question. I suppose one of the big difficulties with studying MS is how symptoms usually come very suddenly, and then there's a very rapid decline in health of the patient, and therefore we're given a very short time frame to analyze MS and figure out its pathology. So trying to figure out where to start, I suppose, when studying this, because my goal was to find something, and I didn't know how to find that something at first. So first of all, taking a step back, looking at my videos just observationally, I found something behavioral rather than just using typical numbers and clinical scores. I found something that is visible and that otherwise you wouldn't be able to find using other tests that only give you numbers on a graph. By searching the number of breaks in the way that they accumulate over time, it's something that was more difficult to find, but once we got it, I feel like it gave quite a substantial result. I hope that answers the question well enough. But
thank you. I'm sure it did. It's so hard when you don't have audience feedback. So I also just wanted to highlight a comment that was made. So happy to see Aaron and Martin highlighting a medico and demonstrate the qualities of the community they are building. So wanted to point that out, that we have people from all different organizations in the crowd listening to this, and I think it's reflective of the mandate of Campus Alberta Neuroscience. With only 1 minute left before we take a 10-minute break, I'm wondering if we could have a quick piece of advice from your perspective, both Jordan and Dr. Ferguson, starting with you, Dr. Ferguson.
Piece of advice for whom?
Piece of advice for anyone who wants to get into the, let's call it your space specifically, so making community health impact.
Yeah, I think about your values and your priorities, and if you can align those values and priorities with the challenges that we're seeing and expecting to see in an aging population over the next 20 years, if you see that as something that is really potentially rewarding for you, go for it. It's an exciting space to be in and an impactful space to be in.
Wonderful, thank you. Jordan, how about yourself? Let's call it in the context of advice to fellow scientists.
Absolutely. I'm, as an undergraduate student, I guess advice to other undergraduate students is contact professors and see if you can get involved in the lab. It's quite intimidating the first time, but professors are usually very polite in answering, and you can make some wonderful connections and have excellent experiences that just are so enriching to your experience at university. I highly recommend.
Well, I love that. And on that positive note, let's take a 10-minute break and regroup for our next speaker, which will be another trainee spotlight. So I will see you all in 10 minutes. I'm going to put my screen off and I'll be back online in about 8 minutes. Okay, welcome back, everybody. I hope that was enough time for a quick break. Excited to almost kick off the almost afternoon. It's 11:20. Is Katelyn Shewchuk. She is going to be our trainee spotlight, and I will let her take it away for the next 10 minutes.
Awesome. Thank you so much, Crystal. I'll just share my screen here. Great. Okay, well, hello everyone. Thank you so much for having me. I am a PhD candidate in Dr. Sarah McFarlane's lab, and I'm really excited to talk to you about some of the work that I've been doing for my project titled Semaphorins in Mueller-Glia-Directed Repair. Sorry, 1 second. So just for a little bit of background, the retina is a tissue found at the back of the eye, and it's responsible for vision. Upon closer look, we can see that the retina is highly organized into distinct layers, which contain a myriad of neuronal cell types which make up its complex circuitry. Retinal degeneration is characterized by the loss of retinal cells. So here is a healthy retina. And here are two examples of retinas experiencing retinal degenerative disease. So up here we have an example of age-related macular degeneration, and these yellow patches are where photoreceptors have died. Down here we see an example of retinitis pigmentosa, and these pigment deposits are where other retinal cells have died. Upon the loss of retinal cells, there's a disruption of the circuitry, which leads to subsequent vision loss. Unfortunately, the mammalian retina does not have any endogenous mechanisms that allow for the regeneration of the retina, and so subsequent vision loss is permanent. And what happens rather is we go from having this healthy retina to a retina that looks a little bit like this, and the formation of what is called a glial scar that looks like this. And so with permanent vision loss being a significant burden for patients, there's been a lot of research geared towards trying to find ways to reverse this degeneration, such as stem cell transplants or intravitreal injections. However, there are some barriers or many barriers to these different options. So a lot of us are turning to an animal model that's able to regenerate its retina on its own. And so one of the main purposes of my talk today is to emphasize the power of the zebrafish model in understanding regeneration, as it is known to regenerate many of its tissues, including the retina. And so I'm particularly interested in understanding how zebrafish regenerate their own retina. And not only are we interested in them because of their regenerative capacity, but the zebrafish have high conservation with retinal structure and genome to humans. And so when we're trying to understand physiological processes, this can be very relevant when we're trying to understand or inform new treatments. Additionally, zebrafish are highly amenable to genetic manipulation, and so that allows us to generate new ways to look at the way that genes and different signaling mechanisms are involved in different processes by generating loss of function models or gain of function models. And so zebrafish are able to regenerate the retina through a specific cell type called Mueller-Glia. So this is a cross-section of a zebrafish retina right here. You can see the photoreceptor cells here in red, which are responsible for detecting light. And then in this layer called the internuclear layer, we see the green Mueller-Glia cells here. And so their cell bodies are in the internuclear layer, and their processes span throughout the entirety of the retina. And so Mueller-Glia are found in all vertebrate retinas, and they're responsible for maintaining homeostasis within the retina and supporting neuronal health. Additionally, in all vertebrate retinas, they respond to injury. However, the way that they respond to injury differs between species. And so in the zebrafish retina, Mueller-Glia are highly neurogenic. They produce rod photoreceptor cells, and then in response to injury, they're able to regenerate any lost retinal cell type. However, in the mammalian retina, Mueller-Glia produce that glial scar that I showed you earlier, and they do not contribute to regeneration. And so the field is really interested in understanding why do these regenerative differences exist between species. And it is believed that signaling mechanisms in the cellular environment are what drive these differences. And so I'm going to quickly describe to you what Mueller-Glia-driven repair looks like. So we have our resting Mueller-Glia here, and upon injury, there is a reprogramming event that allows for the Mueller-Glia to undergo an asymmetric division to produce a single progenitor cell. This progenitor rapidly proliferates, and then these progenitors are able to differentiate into any retinal cell type and restore the circuitry. And so this process has been investigated in the literature, and there have been many genes identified that regulate these different steps. However, we do have a very long way to go to fully understand this process, and that's where my research comes in. I'm really interested in understanding genes that regulate the resting state of Mueller glia and how they're able to become activated and then return to a resting state. And so that brings me to my protein of interest, which is semaphorin 3F. Semaphorin 3F is a secreted molecule traditionally associated with axon guidance. However, more recently, it's been associated with cellular processes that are crucial for regeneration, such as cell survival, proliferation, and migration. Additionally, within the lab, we've identified that semaphorin 3FA, which is the zebrafish ortholog for SEMA3F, is expressed within Mueller glia. So this is a cross-section of a zebrafish retina again, and we performed a fluorescent in situ hybridization for SEMA3FA. So that labels SEMA3FA mRNA transcripts. And so those transcripts are these green puncta here. And then I also labeled Mueller glia in orange here via glutamine synthetase. And so we can see a nice co-localization of the expression of SEMA3FA by Mueller glia. I additionally found that there is a change of expression of Mueller glia with an injured versus uninjured retina, and it is in fact downregulated in the eye. So again, what I did is I performed an in situ hybridization for SEMA3FA in zebrafish in uninjured and injured retina. And so this is what that purple stain is. And so at 48 hours post-injury in our age-matched control, we see nice expression of SEMA3FA in the internuclear layer, which is where Mueller glia reside. And then in the injured eye, we see a nice downregulation. I was also able to measure this downregulation via qPCR between our injured and uninjured eyes at 24 to 96 hours post-injury. So we do know that there is a downregulation of its expression. And so when we see this change in expression, that indicates to us that SEMA3FA is somehow participating in the regulation of regeneration by Mueller glia. And so that brings me to the focus of my project, which is to characterize the role of SEMA3FA in Mueller glia function in the zebrafish injured retina. And I want to bring us back again to really emphasize the power of zebrafish and their genetic amenability. We are able to generate really quickly loss of function or even cell-specific gain of function models, and I use both within my project. And I'm happy to talk about it a little bit more later, but for the purposes of this talk, I'm going to be telling you about my loss of function data. And so the way that we generated this line was through CRISPR-Cas9, and this is a transgenic zebrafish line that is a whole body knockout for SEMA3FA. So these zebrafish actually do not express the SEMA3FA protein at all. And so I want to use these mutants or these loss of function mutants to see how is the regeneration process different than their wild type or normal siblings. And so I want to analyze 3 different steps of the regenerative process in our mutants. I want to first look at the proliferative response. I then want to see, is there any changes in gene expression of genes associated with regeneration in our mutants? And then I want to also look at the overall recovery of my fish. But for the purposes of this presentation, I'm just going to be showing you some of my proliferation data. And so what we use in the lab to understand proliferation is something called EDU. EDU labels proliferating cells. And so what the experimental model looks like is we injure our fish at 5 days post-injury to activate the Mueller glia and get them to start proliferating to produce those progenitors. I then put the fish in an 8-hour EDU pulse, which allows for the incorporation of that EDU into the cells, and then I process the tissue at chosen time points. And the way that we analyze this data is that we see the EDU positive cells here in red, and we count the number of EDU positive cells in each section between our mutants and our wild type injured retina. And so here's data from 72 hours post-injury, and we counted the number of EDU positive cells in the internuclear layer here, as you can see. And so what we find is that when we lose SEMA3FA, there is a stark increase in the percent of EDU positive cells found in the internuclear layer, which, if you remember, this is where Mueller glia reside, compared to their wild type injured siblings. And so there seems to be a prolonged response when we lose SEMA3FA, which implicates SEMA3FA in regulating or potentially inhibiting proliferation. And so that kind of aligns with our data because that's why potentially SEMA3FA is expressed in our resting Mueller glia, and then upon injury, it becomes downregulated, allowing for that proliferation, and then at some point becomes upregulated. And because in our mutants, there's no chance for it to be upregulated, that proliferation is prolonged. And so these findings actually align with the literature. SEMA3F inhibits the proliferation of cancer cells, and it does interact with multiple pathways associated with quiescence and activation. And so I wanted to be able to see if I could quantify the number of resting Mueller glia in our mutants versus our wild types, but that's actually a lot easier said than done because there's no actual marker for quiescence. And so we turned to a really interesting tool generated in another lab that measures, so it's a live tissue cell cycle biosensor that allows for the identification of different cell cycle steps of a cell. And so I'm happy to talk about this at the end of the presentation, but all you need to know is that depending on the localization of the different components within the cell, we can identify which cell cycle stage the cell is in. And so when I'm looking at this compared to our mutants and our wild types, I want to be able to count the number of Mueller glia that are in a resting state or potentially in G2 or even proliferating between our mutants and our wild types. And so I'm taking this biosensor and editing it to make that it is expressed in Mueller glia only, and then we'll be able to visualize proliferating cells and their activity within live tissue, which has never been done before. And so just to summarize what I've showed you today, I implicated SEMA3FA in Mueller glia regeneration. And I do just kind of want to step back again and share and talk about how powerful the zebrafish model is in understanding regeneration. We're not the only group that are looking at zebrafish. There are people looking at heart regeneration, spinal cord regeneration, and they're just so powerful because there are many tools that are available to us to be able to generate new transgenic lines, but also to visualize things that have never been able to be visualized before. And so hopefully these findings will inform us of potential new treatments such as gene therapies to reverse degeneration in the future. And so I just wanted to say thank you so much to my team, my wonderful supervisor, and my funding sources, Spinal Cord Nerve Injury and Pain from the HBI, as well as Hotchkiss Brain Institute. So thank you so much for listening.
Thank you, Katelyn. You spurred some interest in the chat, which we will save for later on. And it's along the lines of, can humans adopt all of these zebrafish powers? I sure hope so, because that is an inspiring research that you're doing. And as you alluded to, there could be a lot of transferability that would make huge human impact. So more on that in a couple of sessions, because we will welcome you back for that roundtable discussion. Thanks again, Katelyn.
Thank you so much.
And next up, we get to speak with Dr. Gerlinde Metz from the University of Lethbridge, coming all the way from LA on the Zoom room here. Beautiful background, by the way. I know you were adjusting the light and it looks perfect. I'll let you introduce yourself, Dr. Metz. You have an incredible background, very diverse. I'm excited to hear more about it. And then after you introduce yourself, you will present your session on using metabolomic biomarkers to improve quality of life for patients with neurological diseases. So here we go. Thank you.
Thank you so very much, Crystal, for your kind introduction, and special thanks to the organizing committee for inviting me to be here today. It's really a pleasure to be here and talk about metabolomic biomarkers to improve quality of life for patients who are affected by neurological diseases. And I'll give you a few examples of how we're using our technology. So I'm at the Canadian Centre for Behavioral Neuroscience, but in order to better understand recovery of function and what neuronal injuries are doing to our recovery and how they're affecting our long-term outcomes, we're also interested in studying biomarkers. So I'd like to start to talk about the rationale why we're using biomarkers in a clinical scenario with talking about traumatic brain injury. So mild traumatic brain injury or concussion may cause a variety of symptoms, including loss of consciousness, memory loss, balance problems, and many others. And these symptoms can occur only for a short period of time, or the onset can be delayed. So this makes it very difficult to have a clear diagnosis for the occurrence of a traumatic brain injury or concussion in particular in this case. So this is why about 5 out of 10 concussions often go undetected. Although there are many diagnostic technologies and treatment strategies available, we're trying to improve the diagnosis of and the chances for a successful diagnosis of mild traumatic brain injury. The challenge is that often when a patient seeks a physician's help, the diagnosis is often based on neurological interpretation of neurological symptoms, and there's no clear gold standard metrics for how to evaluate the injury and how to objectively estimate the prognosis and the outcomes and symptoms. So we are really in need of a more reliable objective diagnostic tool that can also help us monitor recovery of these symptoms over time. So better detection of the symptoms would ultimately translate to better treatment, more successful treatment, and better outcomes for the patient. So this is what drives us. And we are really interested in looking at the complex interactions of what a neurological disease, how it trickles down to affecting functional outcomes. Here you can see a pathway, how a neurological or injury or a damage to neurons or stress associated with it can affect epigenetic regulation of gene expression patterns that will affect physiological measurements and ultimately also affect metabolic changes at the cellular level. And the metabolic changes at the cellular level are actually downstream that directly are reflected in functional changes, because if an organ, due to altered metabolic function of cells, if the organ doesn't work well, like the brain, we will have functional impairments. And the question is, how can we better assess this and help clinicians gain a better diagnostic tool? So there are different approaches of how we can assess what's happening after a neurological injury or a chronic disease, including assessing DNA expression changes at the genomics level, then transcriptomics, or assessing protein downstream with proteomics. And we're looking at metabolomics here, which includes the study of metabolic changes at the biochemical level. So metabolomic biomarker is really a marker that is based on molecular changes at the cellular level that is directly affecting the biological phenotype. So the metabolic changes result from pathophysiological processes resulting from an injury, and these markers can then help us with early diagnosis and more robust diagnosis, but they can also assist in predicting outcomes and monitoring recovery over time, and also do risk prediction in terms of chronic diseases or chronic toxic stress exposures to predict what kind of other health risks or comorbidities might be coming down, might be expected in the future. These metabolomic technologies also enable targeted biomarker detection, but because we can study these omics markers in a very broad sense, we can also use them for biomarker discovery of new markers and therapeutic targets. So we're very fortunate at the University of Lethbridge to have a really high throughput, top-notch metabolomics platform available, which is directed by Tony Montina. And we have developed a long-term collaboration over the past 10 years to study a variety of conditions. And I'd like to show you a few examples of our collaborative projects. So on the right-hand side here, you see a proton NMR spectrum that we get, and it shows the resonances and sets of resonances or signals that can be attributed to specific metabolites in the sample. And you can use actually very clinically accessible samples such as urine or blood or saliva. And this makes it very interesting because these samples can be even collected at home. We can go pick them up and we can monitor changes in metabolic profiles, personal metabolic signatures over time. So these approaches have also led to patent applications coming from our platform. So here I'm showing you one of the samples of our recent studies. This has been really wonderful collaboration with Dr. Chantal de Bird at the University of Calgary at the Foothills Hospital. And she's been working with a group of varsity athletes who provided her with urine samples at the beginning of the training season, and then they provided her with another set of urine samples at the time in case they had to visit the clinic with a concussion that had occurred due to their training and physical activities. So what you see here on the left-hand side is actually what we call a partial least square discriminant analysis course plot. But in simple terms, it shows the separation between specific pre-injury metabolic signatures in blue and post-concussion metabolic signatures in red in the urine samples. The symbols in the ellipses indicate individual samples and shows where they were clustered. And you can see that the post-injury metabolic profiles were significantly different from pre-injury profiles. And the specific metabolic signatures, very personal signatures linked to the presence of concussion, included a small number of specific metabolites, including phenylacetylglycine and biotin and heparin and so on. So we were also curious of what happens over time. I was mentioning we're interested in recovery of function. What happens over time to those metabolic signatures? And here's what we saw after 6 months of recovery time post concussion. So these are the samples that we looked at before. You had the initial samples, metabolic profiles in red here, and then the post-recovery samples in blue on the right-hand side. And the metabolic profiles between these two time points were clearly different, very significant differences. And we found that particular metabolites linked to the length of recovery included hydroxybutyrate, and there's a marker for symptom severity that was lactose. So we were, through machine learning analysis, identifying these particular metabolites that can give us a very personal profile of this particular concussion and how the symptoms are moving forward over time. So I've switched gears a little bit because we talked about an acute neuronal injury here, but what about neurodegenerative diseases where the progression of pathophysiological symptoms occurs over an extended period of time? So I would like to point out a study that we have done on Alzheimer's disease. And what is really peculiar about Alzheimer's disease is that often the neuronal loss can occur 25 to 30 years before actually symptoms emerge and before this disease can be diagnosed in the clinical scenario. So it would be very helpful to have a marker that can help us with early diagnosis or predicting risk and giving a prognosis for future development of the symptoms. So we were actually very fortunate to work with Dr. Jeff Joseph, a pathophysiologist and pathologist at the University of Calgary, who provided us with brain samples post-mortem from patients who have been affected by Alzheimer's disease. We received from him 8 different brain regions, and we did metabolomic analysis, again, with Tony Montina in a collaboration with a postdoctoral fellow in our lab, Mirela Beskovic. And these 8 different brain regions showed a very interesting pattern of metabolic changes that was very specific for each of these brain areas. So what you are seeing here is a heat map across these 8 different areas we looked at that shows the predominant metabolic regulation for each of these regions. Red indicates that there was a general downregulation of metabolic changes, whereas green indicates a general upregulation of metabolic pathways. And you can see that the most severely affected regions included the frontal cortex, which is BA9, the temporal cortex, and parietal cortex. And what's really interesting here is that the metabolic changes reflect the neurodegenerative progression of the disease. Now, we looked at particular pathways and metabolites that were altered, and the bar graph here shows you the actual number of metabolites that were altered in each of the brain regions. And you see that the largest change occurred in the BA9 region, the frontal cortex, yet again. So the metabolites that were found to be significantly affected in this case, most changes occurred in phenylalanine metabolism and GABA. Both of them are actually related to neurotransmitter production. So this again gives us a very good link to the functional symptoms of the disease. And now we're trying to use more accessible samples like urine or blood or saliva to identify markers that can be provided and studied in a clinical environment. So to summarize these findings, biomarkers in clinical practice are very helpful for the study of a signature. Multiple markers can be used in metabolomics, which I think are more robust in their results compared to a single biomarker, more reliable, and they provide high accuracy for a diagnosis. They can enable an objective assessment of symptom burden and recovery. They're cost-effective in this case of metabolomics and can be done on accessible fluids for personal management and decision-making of the treatment pathway. I'd like to give you a quick insight into some examples of how we're applying these biomarkers on a day-to-day basis. For example, we're assessing a therapeutic success over time in a virtual reality intervention to enhance cognitive function after traumatic brain injury in collaboration with a corporate partner, L47, run by David Tinjust. We are also interested in monitoring the success of a power chair soccer intervention for individuals with physical disabilities. This is a program run by Chase Petruska in my lab and working with Tony Montina. We are also working on risk prediction of disease, for example, working with populations and individuals exposed to extreme stress, such as NASA astronauts living on the International Space Station, or populations exposed to violent conflicts or natural disasters like wildfires and floods. These are very close to home studies in collaboration with Afshin Beheshti and David Olson and many others who are providing us with access to a lot of these established population cohort studies around the world. So I would like to thank my very hardworking team, my great students, my fantastic collaborators around the world, and also the funding agencies for their support. And last but not least, thank you for tuning in and for listening, and I'd be happy to take any questions. Thanks so much.
Thank you, Dr. Metz, and we do have just a few minutes of questions. I particularly like your focus on precision medicine and personalized approach. That is the way of the future. It's what everybody's asking for, and nice to see that happening at the early stages in the research lab. So appreciate that. Curious, as you're looking at all of the ways that you can make impact in Alzheimer's disease, how are you feeling about moving this research so that it gets into the community at a large scale?
Yeah, this is really close to our heart because ultimately we really want to make a difference at the patient side and affected populations. So we're running maybe proof of principle studies, you would say, with smaller cohorts, with smaller studies. And we're able to collect, for example, urine from individuals affected by various conditions. We can actually ask them to collect their urine at home. And we are going, right now, we have students driving all across the province to go collect these samples from individuals who are actually not able to bring them to us or to even go visit a clinic. I really enjoyed Dr. Ferguson's talk about the rural communities and barriers of accessibility. And this is a big problem, but I think with some of these easy to collect samples, we can help improve the ways how we interact with patients who don't have the mobility to seek a clinical environment and get a blood sample taken, for example.
Love the patient-centric mindset and approach. Now we have one quick question, Dr. Brian Kolb. What about factors such as sex and age?
Oh, this is a great question. Thank you so much, Brian. This is a wonderful consideration because we do need to take into account sex and age factors. We're studying these. Usually it's we try to have a matched population, and we do see very significant sex differences. But there are challenges when I talk, for example, about the study with NASA and astronauts. We do not have very female astronauts, but we were actually even able to study sex differences there with a small number of female astronauts. And what we have shown in this example is that females do adapt very differently to stress over time. So it is highlighting the need to consider sex and age differences in all of our studies. Very, very important.
And going back to that precision medicine personalization.
Exactly.
Love that. Well, I'm sure there will be more questions to be had in the round table. But first, we do have one more speaker. So thank you, Dr. Metz, and we will see you soon. Please stay online. Okay, next up we have Dr. Gibb, Robbin Gibb. I'll let you introduce yourself. Would love to hear more about your background, how you became interested in this world and what the pathway has looked like, and then your presentation on using tactile stimulation as a means to promote recovery after early brain injury.
Okay, well, thank you. And thank you to the organizers for inviting me. I have to admit, I was a bit surprised to be invited to a recovery after brain injury symposium because it's been a while since I put my hand to looking at these sorts of things. But I am a fierce proponent of tactile stimulation and what it can do for both the normal brain and the injured brain. And so I'm excited to be able to share some of the findings that we have with you. Before I get started, I hear Brian Kolb is one of the participants here, and I'd like to acknowledge that when I did my graduate work with Brian, he allowed me to decide what it was I wanted to work on. And in the early 80s, he attended a conference in Israel and came back with this notion that had heard from Dr. Tiffany Field there, that tactile stimulation seemed to make a difference for promoting recovery in babies who were born preterm and allowing them to be released from hospital sooner. And because we were at the time working on models of brain injury, Brian suggested that we try it with our rodent model, the rat, after a postnatal day 3 injury, which is pretty devastating for a rat. And that's really where it all began. So I started my interest in tactile stimulation when I was working with Brian, and that hasn't gone away. So I think there's a lot to be said for using these experiential approaches to make a difference and help promote recovery after a brain injury. So I wanted to start by talking a little bit about the skin. It is our largest sensory organ, and it actually helps us determine when we're being touched. We get a sense of pressure, and we get a sense of temperature from our skin. And there's other things as well, including itch, but it really is our biggest sensory organ, and it does a lot of our sensations for us. It helps us understand what is painful and what is pleasurable as far as interacting with our environment goes. And it does provide a barrier and a medium to interact with our environment. So it is what separates us from the rest of the world. It helps us regulate our body temperature. And what I found was interesting about skin is it's continually being replaced. We have a complete turnover in about 1 month, and it accounts for about 6 pounds of our body weight. We lose about 9 pounds of skin per year, so that's about 3/4 of a pound each month. Oops. Okay, so the next point I want to make is it appears that something that I wasn't top of mind when we started doing these studies is how closely related the brain and skin are. So embryologically, they're both formed from the ectoderm. If you look at the components of both the brain and the skin, they have stem cells, and they produce and utilize similar proteins for reparative processes. And this is when I really became interested in what the skin could do as far as helping heal a brain. So some of the proteins that make a difference for this repair process in the brain include fibroblast growth factor 2, nerve growth factor, epidermal growth factor, insulin-like growth factor. All of these have been demonstrated to make a difference for repair in the brain, and they also help with repair in the skin. Now, when I started looking at the literature to try to understand why it was that tactile stimulation may make a difference in brain recovery, I ran across a paper that was written in the 70s, I believe, and it was talking about how brain homogenate had been used to successfully treat very severe burns and other skin lesions that were refractive to other treatments. And again, it got me thinking along the lines that perhaps these things are so closely related, we have underestimated the potential for skin to make a difference in recovery of brain injury. In about 2015 was a paper published by Lloyd. Francis McGlone is on this paper, and they really described the neurobiological pathways of touch to include a C-fiber somatosensory pathway that runs through the body that has never previously been discussed. So we know that we have these C-fibers, and they have classically been thought to transmit pain and itch sensation to the brain. But it appears that there's also fibers within that system that are sensitive to pleasurable touch, such as massage. So this is the C-tactile fiber system, or CTs. Now, these fiber systems actually stimulate regions in the brain, including the frontal cortex, prefrontal cortex, and the insula. And both are involved in emotion, both are involved in relationship. So it's interesting that we now know that we have this ability in our skin to detect things such as pleasurable touch. And it looks as though that sort of fiber system may help promote emotional, behavioral, and hormonal responses to touch. So the neurobiological pathways of touch have been demonstrated as well. And this is a paper that was written in 2017 that talks about how we form pair bonds through the action of mu opioid receptors, or MORs, and these are for endogenous endorphins that are produced by the brainstem, and it sort of provides a neurobiology of attachment relationships. So these MORs have a stress-reducing action, and they are responsible for helping us maintain our social bonds throughout our lifespan. A hormone that is produced by touch is oxytocin, and it is produced in the hypothalamus, and it helps regulate emotional states and our stress axis, the HPA axis, as well as the autonomic nervous system. Again, release of oxytocin does have a stress-reducing effect, and it is released in response to these social interactions. Another positive effect of oxytocin is it increases brain plasticity. So the thought that extra early affiliative care, such as infant massage, may be a critical mediator of enhanced regulatory capabilities over stress responses comes to mind when we understand that touch can enhance both social and executive capabilities. So tactile stimulation, as I mentioned previously, was first used to improve recovery after early brain injury or early trauma. And a lot of this was based on Dr. Tiffany Field's work in premature human babies. So we, as I mentioned, Brian Kolb and I were interested to understand whether we could get behavioral recovery or anatomical recovery after early brain injury with tactile stimulation. And to our great delight, we discovered it works. So using a variety of tests, including tests of motor function like tray reaching and tests of spatial navigation like water maze, we were able to demonstrate that we could improve behavioral recovery in animals who had an early brain injury. We also published a paper just recently on hemidecortication, and we were able to show in those animals that they had behavioral recovery as well as some anatomical support to anatomical changes to support that behavioral recovery. So one thing I thought was very interesting by the work that we did in these early days was that often when we look at various treatments for brain injury, we have a hard time reinstating motor recovery or sort of stimulating motor recovery. And yet tactile stimulation seems to be a wonderful way to do that. So our animals that are usually refractive to changes in motor recovery, we often see cognitive recovery, but not necessarily motor recovery. They, with tactile stimulation, actually demonstrated a better behavioral recovery. So when we talk about animals, a lot of people who are probably tuned into this may wonder, well, that's wonderful for understanding how you can help a rat recover from a brain injury, but why would we care about that as far as humans go? And it turns out that rats are actually a very good model of human health. 90% of their genome matches the human genome, and of the remaining 10%, about 9% has homologous genes with the human genome. Rats are clever. They're easy to work with. I absolutely love working with rats as a lab species. They have a short lifespan, high rates of reproduction. They have fairly large litters, and we can control their experiences. So this is one of the key reasons that rat models actually are wonderful to use while we're trying to understand how it is that that things like tactile stimulation can make a difference. We can map experiences to alterations in the epigenome or even neuronal morphology. There are some differences between rats and humans outside of our size and our fur. There's no tonsils or gallbladders. Rats do break down toxins more efficiently than humans, and there are some differences in genes that code for immune and olfactory systems. But I wanted to bring your attention to work that was done by Michael Meany, and this was done decades ago now, but he looked at how two different classes of rats, high-licking grooming mothers versus low-licking grooming mothers, can make a difference for outcomes in their babies. And this little visual is one I like to use to demonstrate that when you have a high-licking grooming mom shown here with the tan color, her babies tend to turn out to be high-licking grooming animals as well. And when you put them in a stressful situation as adults, they have they're able to manage that situation. They have a low anxiety response, and ultimately their cortisol levels drop relatively quickly, and they show more resistance to things like dementia later on in life. Now, these white animals are low-licking grooming moms, and they tend to give birth to low-licking grooming babies when they grow up will be low-licking grooming moms. You put these kinds of animals in a highly stressful situation, and they have a high stress response. They spike cortisol, and it stays high for long periods of time, it causes some degeneration in both brain and body over time. So these animals are more at risk for things like dementia or early cognitive loss versus these animals. Now, the cross-fostering experiments here with licking and grooming, with a mother who gave birth to low-licking grooming pups given to a high-licking grooming mom, she can actually convert them into high-licking grooming animals, and they show this lowered response or anxiety response in a stressful situation. And then when you take pups from a high-licking grooming mom, give them to a low-licking grooming mom, those pups will grow to be high-licking grooming moms as well. So it's almost like there's a safety net built in. Just having been raised by a low-licking grooming mom was not enough to change their behavioral phenotype into low-licking grooming animals. So this work shows us that even in the intact brain, a lot of tactile stimulation early in life is beneficial. So in the lab, we have converted our work from using little paintbrushes to actually using Swiffer dusters, because we can stimulate an entire litter early on in life with these Swiffer dusters for 15 minutes at a go. We typically use tactile stimulation for 2 bouts of 15 minutes each time. And we are able to show that that we can stimulate the production of these proteins in the skin with tactile stimulation. So this is the FGF2 protein. We see it increased in the skin of the animals that are tactilely stimulated, not in the animals that did not receive tactile stimulation. And we can also see increases in this protein in frontal cortex, parietal cortex as well. And we see changes in receptor for it in parietal cortex. So that's Western blot of skin. That's what FGF looks like. If we actually give FGF to the animals, they show similar behavioral recovery. So we saw that they could make a difference. Tactile stimulation makes a difference in baby rats. We were curious if we could see similar changes in adult rats. And so we actually tried it with adult rats who were given early brain injury. It improved their behavior. It also gave us some indication that we were changing the neural anatomy underlying the behaviors that we saw changed. And we even saw improvements in adult rats with stroke. So in another model, the Alzheimer's model using APP mice, this was work that was done by Brian Kolb and Russell Hussain. They were able to show that it made a difference in these mice as well. So they were more resistant to developing Alzheimer's with tactile stimulation. So we were hoping that we could actually apply this in the human condition, and we have been working on a proposal with Brian Kolb and others at the University of Calgary and University of Edmonton to try to get training to parents who have babies who were born with hypoxic ischemic encephalopathy, and hoping that we can use tactile stimulation to make a difference for their recovery. So that's what I had for you today. Here are my references. I have so many people to thank over the years, but importantly, I would like to thank the funders, the NSERC, who funded a lot of this work, and Campus Alberta Neuroscience, who funded some of it as well. Thank you.
Thank you, Dr. Gibb. What a simple yet complex therapeutic answer to something that is of high relevance and a major issue in the world today. I'm going to use this presentation as a personal justification to paying for monthly massage therapy. But I'll be curious to follow your research to see how you envision it translating. Like, is there going to be a specific type of protocol or type of touch? Are we going to use Swiffers on babies? What do you imagine it'll look like?
Well, actually, I just last year was trained on infant massage. There's an international association for infant massage, and me and a number of students in the lab and other trainees decided we would take the training for this. And we have been going out in the community promoting the notion that infant massage is good because we've seen it makes a difference in normal brains, intact brains, and it looks to make a difference for babies that have issues neurodevelopmentally. So we're hoping that by just bringing up a higher profile on massage itself. And so I do think you are well justified in having those massages, that we can get people to understand that how closely related the skin and brain are, and how you can use the skin to actually cause changes in the brain and for the betterment of life outcomes.
Wow, that's fascinating. And I have it written down that we lose and regenerate 9 pounds of skin a year. Whoa.
I'm surprised at that too.
Yeah, you're full of fun facts. So just like we were talking about transferring from the rat models to babies, in all seriousness, when I'm talking about justifying my massage therapies, I'm obviously not a baby. Do you think that transferability could also apply to a later stage adult who may be facing with anxiety in their future, in their past or present?
Absolutely. In the beginning, we weren't sure, because we weren't sure that the proteins the skin was making would actually cross the blood-brain barrier. When you're in the developmental period, it looks like you have a leakier blood-brain barrier, and so things can cross more easily. So we weren't sure, and that's why we tried that experiment with animals that had been given an early brain injury, but we didn't give them tactile stimulation until they were fully adult, and we saw this beautiful behavioral recovery that was associated with that, and also anatomical changes.
Wow. And is there too much tactile stimulation? So it seems like such a simple therapy that you would think that someone would be like, I'm just gonna.
Yeah. No, I think you have to bear in mind that it should be pleasurable. When you get into a point where it's off-putting or people are resisting the touch, then you have to stop. Because the whole point is that it's supposed to be reducing stress, not causing stress. And clearly you have to keep in mind the emotional tone of what it's producing in the person who's undergoing that tactile stimulation.
Yeah, well, and on that same theme of transferability, and as personally, I'm a dog mom, not a human mom. Do you think that there's a transferability in other animals?
Oh, absolutely. As a matter of fact, I used to work in the lab with somebody who was an animal trainer, and he promoted the notion after the work that Brian and I were doing on tactile stimulation. He promoted the notion that he had better success with pups, people who were coming to him with problems with their pups, if he promoted more tactile stimulation. And so I think breeders are onto it, because I've heard now that they are actually doing more touch with the pups, trying to get them in a better space for human contact when they're finally adopted out.
Oh, I love that. Well, when you increase the awareness to mothers and caretakers of young children and babies, I'm sure using animals like puppies as examples is always an almost like a cheating way to get the word out.
Right. Everybody's interested.
Everybody's interested. Yeah. Well, thank you so much for all of those fun facts. And if you don't mind staying online, I'm going to also bring back Dr. Metz and Katelyn for a roundtable discussion. So thank you, Katelyn, Dr. Metz. What I love about this, including this roundtable and the previous speakers, is there is so much to learn about the brain. Holy, just when you think, it's almost like the more you know, the more you think you know, the more you realize you don't know. It's just this. And I find that of all the scientists that I meet, you're attracted to the unknown. Like, you are all entrepreneurial, you're explorers, and you have this passion that is very contagious. So on that note, I want to start with the same question I did on the previous roundtable discussion, which is why? Why do you do this? I'm sure there's a lot of challenges of being a scientist, but what gets you out of bed and gets you excited about your role as a scientist? Starting with you, Dr. Metz.
Thank you so much, Crystal. Yeah, you put your finger on the soft spot here for us because it's really close to our heart. Ultimately, we want to make a difference to the community. And that's also what Dr. Gibb was alluding to, that we really have to bring all our animal research out there to the community. We have to do proof of principle studies in population-based studies or clinical cohorts. And we want to make a difference. There's no point in doing research for fun in the lab if it doesn't help and give back to the community. And so this is what's driving us. And actually, I am always talking about reverse knowledge translation, because we're not only translating the knowledge from the lab to the real world, but the real world gives us the questions that we need to bring back to the lab in order to do meaningful work. And that's really important. So we're asking the relevant questions, and we're learning about mechanisms, and then those mechanisms will inform new treatments or biomarkers and things like that.
Yeah, that bidirectional communication, that I love that. Reverse knowledge translation. So important to have that voice and infinite curiosity. How about yourself, Katelyn?
I mean, Dr. Metz kind of like encapsulated that, giving back to the community and taking from the community. I also wanted to kind of talk about my experience with science in general. Like, I'm super passionate about understanding mechanisms and solving problems, and that's kind of what drives me coming to the lab late at night. Oh, how do I fix this experiment? How do I, solve this problem? And because I am kind of more of a basic science researcher, I love being at the foundation of generating knowledge to then translate into treatment. So how do we even figure out that this treatment or this pathway could be used in this particular gene therapy or this particular pharmaceutical? So just really understanding how the mechanisms work, that's what really drives me. And I love working with different animal models. I think I've worked with mice and zebrafish, and they're so powerful in understanding the difference between different species and things. So that's kind of what drives me to come to the lab every day.
I really appreciate those comments, Katelyn, because something I admire about scientists, especially scientists who are working with animal models and at that basic science research, is you have incredible patience. You undoubtedly want to see that community impact, but you know the importance of foundational research so that this is scientifically valid information and therapies that are getting out to the community, that it's been tested and gone through the scientific rigor. So there's a reason why my foundation, the Branch Out Neurological Foundation, as much as we love the innovation and getting research beyond the lab, we have 50% of our funding goes towards basic science for that reason. So really appreciate those comments. Now, how about you, Dr. Gibb?
Well, I have to admit that I came to science sort of later in my life. I worked as for a long time with Brian Kolb as a lab technician while my children were growing up. But definitely when I had the opportunity to ask the questions, it was driven a lot by what I saw in my own children and now grandchildren, wondering how we can make the world a better place for them. So a lot of what I do is driven by the understanding that we need to leave our world in a better place for the next generation coming along. And I think the more we understand the simple approaches to making a difference for outcomes after brain injury, the kinds of things that happen inevitably in life, those kinds of things, I think, understanding a little bit more about what we can all do in very simple terms to make a difference. That's what drives me. I'm really interested in how everyone can contribute to making better outcomes for others.
Well, that certainly shines through with all of the work that you're doing. Now, I have a few more questions. We've got 10 more minutes here. I have a couple of specific questions for you, Katelyn, from the audience, Ilya, Rashid. And then I'm going to turn it on the three of you. I'm going to ask you, Dr. Gerlinde Metz, to ask Katelyn a question. Katelyn, I'm going to have you ask Dr. Gibb a question, and Dr. Gibb, I'm going to have you ask Dr. Metz a question. So first, I have a couple for you, Katelyn. The question is, do you think applying the zebrafish ability of regeneration to a human would allow us to give the ability of regeneration to our species?
Yeah, so I feel like there's a lot to unpack there. I think that's the whole purpose of trying to understand all of these mechanisms in the zebrafish. And like I mentioned in my talk, there's high conservation between a lot of the different mechanisms that occur between species. And what we think right now is somewhere in evolution that regenerative ability was lost, coming from zebrafish to mammals. And so there are some groups that are looking at different genetic mechanisms such as promoters or enhancers, and potentially are there some genes that have been silenced in our own biology, and we want to try and awaken those silenced genes in some sort of gene therapy or pharmacological agent to encourage our own retina or our own tissue to regenerate, because stem cell transplants haven't really had much success because the grafts typically are rejected, and they don't integrate into the circuitry in a manner that actually regenerates or restores those functions. And so if we're able to stimulate endogenous mechanisms, potentially that can improve outcomes. So absolutely, I think that's the whole goal of all this research, is to try and apply these findings to future treatment.
On a similar theme and related to this question as a follow-on, do you think by studying gene expression in retinal cells and using gene editing technologies such as CRISPR, ZFN, will also allow us to understanding regeneration of the human eye?
Yeah, so I think that's kind of like two components. I mean, we can't really use gene editing technology in humans for experiments, and that's why we use our animals. But I think the hope is to potentially create a gene therapy that uses CRISPR technology to potentially either edit a gene, activate it, or inhibit something that's potentially inhibiting proliferation to promote regeneration. And that's pretty much the hope. through the gene editing pathway that way. And then obviously we use gene editing all the time in our animal models. I mentioned I use CRISPR-Cas9 to generate our loss of function, but we also use overexpression models and things like that through transgenesis. So yes, all of these are very, very useful to understanding mechanisms and informing future treatment.
Great. Thank you so much. And Dr. Metz, do you have a question for Katelyn?
Oh, Katelyn, I really enjoyed your presentation. Thank you so much. The biosensor topic was fantastic, and there are so many applications. So it made me wonder of how you see applications for someone who's younger versus someone who's at older age. Do you have an idea of what potential opportunities would be in this sense?
Like, so you're talking about maybe a congenital condition versus like aging or degeneration?
That's right.
Yeah, so that's tough to answer. I feel like probably there are some studies looking at Mueller-Glia regeneration in the aging eye of the fish versus like congenital conditions, or there are certain transgenic lines that generate chronic degeneration themselves. And we do find that the regenerative potential within the zebrafish actually stays consistent between the two. And so, you know, I think it would be a pretty similar approach where we would give a gene therapy either early on in life to then change the phenotype or the activity of Mueller-Glia to continuously help to regenerate the eye through a patient's lifespan, or if a patient either experiences an injury or some sort of, because retinal detachment is quite prevalent from head trauma or a retinal degenerative disease, we would then just apply the treatment later on. But that's kind of, we're still in the nascent stages of trying to understand the differences, but you know, retinal regeneration in the zebrafish stays consistent in younger versus older models. So I would think that the treatment would stay consistent as well.
Great.
Thank you for your question.
Now, Katelyn, do you have a question for Dr. Gibb?
Yeah, so I mean, this is kind of coming from a young female scientist to a more experienced female scientist, and I like to take the opportunity to ask these questions when I can. What made you, so like you, I'm like trying to think of how to word this, but what made you want to, so are you, made you want to start your own lab? So you were working with Dr. Kolb as a research associate or a lab technician, and then you decided to start your own lab. And can you like describe that journey? Because I feel like in young academics right now, it feels like it's a race, and we're all really stressed about how we're going to get to where we want to be. And I would love to hear your thoughts and advice as an aspiring academic.
All right. Well, let me tell you that when I finished university here at the University of Lethbridge, I decided to go into graduate work, and I went to the University of Alberta. So I was doing my master's there, and things were going swimmingly well. And so my supervisor convinced me that I should consider doing a PhD. So I switched into a PhD program. And about the same time, my boyfriend, who I started dating in college, said, you know, you should come home and we should get married because I'm sick of driving to Edmonton. And I thought that sounded like a better idea than finishing my PhD. So I stopped doing that work and had a family. And then when my kids were old enough to go to kindergarten, that's when I went back and started working with Brian. And so I had the science sort of never left me. I was always interested in the work and fascinating kinds of things that Brian was doing in the lab. And so when my children graduated from high school, my husband said, you should go back and work on your graduate degree. And I thought, well, it's kind of a long time since I started, but I realized that he recognized that it was something that I needed to do for myself. And so with his encouragement, I decided to investigate the possibility of going back and getting my master's and PhD in neuroscience. And that's what I did with Brian. So it worked out well. I'm glad I did it. It's been fun. I love my work. I love being able to be the one that decides what we're going to put resources to. I like working with students and having students interested in the work we do. But yeah, I find that it's been much more fulfilling than I could have ever imagined. So I'm glad I did it. And so I don't think you have to get in a real rush about it. You can take your time and you can still have impact, I think.
Love that. And lots more to come. Dr. Gibb, do you have a question for Dr. Metz?
Oh, I do. Gerlinde, you do so many things. Spinal repair, MS research, all of the work that you've done on transgenerational influences of experience and stress, and now metabolomics. Tell me what you think is going to have the biggest impact on human health from your own research perspective, all of the work you've done.
Oh, that's a great question, Robbin. Thank you so much. And by the way, I'm so glad you're here and you took this bumpy road to being here and being 2 doors down from me right now. It's just a pleasure to have you here. So, and a very good question. What I think makes the biggest impact, I think, how we're working and having this background in behavioral neuroscience and neuroscience in general, it is so multidisciplinary. And I think that is what we all enjoy so much, being in this field, that we work with so many disciplines. And behavioral neuroscience in particular brings it all together, because whatever we do at the molecular level, whatever hormones we're changing, whatever therapy we're doing, we're looking for a change in behavior. We're looking for better movement, better learning. Something needs to get better in behavior. Ultimately, that is integrating everything else that's going on. And I think that's where we're coming from, studying so many levels, molecular levels, physiological levels, aging, sex differences, you name it. So the biggest impact on human kind or a human population or the community, I think, is really looking at the big picture and doing something that matters to the individual, but also to the community, to integrate what we've learned, not to look at one protein or one particular measure, but look at every person, but also the specifics of a population that is in need, that is highly vulnerable. We have so many vulnerable populations in Alberta, in Canada, that need help. And they all have specific needs, but I think it's important to really bring everyone around the table, do what we call integrated knowledge translation, to really bring everyone together to make a difference to the current problems, to the problems we have in our society. It's about collaboration, really, and good teamwork in the end.
What inspiring closing thoughts. Thank you to the 3 of you and to all of our previous presenters. This has been an incredible day so far. I'm glad we were all able to come together, and it's an honor to be your host. It's a good reminder how far from a neuroscientist I am. My mind is blown. I have so many questions. There were so many words that I did not understand, but it was also a reminder of why I am such a strong neuroscientist groupie. You remind me of the Olympic athletes I used to train with as an athlete, and you remind me of the founders of companies and entrepreneurs that I work with daily. So thank you for all of your work. It's so impactful. You all have that mindset of wanting to make that big community impact, and it's what drives you all, which is very inspiring. Thanks again to all of our speakers, for everyone who attended today. This is a part of having that collaborative spirit that we're trying to ignite, adopting that infinite mindset and actually acting on and asking questions, engaging with unlike-minded experts from different fields in neuroscience. So specifically on the Campus Alberta Neuroscience team. We heard from Neil Neary, the executive director. Thank you. Renee Dumas is on the team. Jackie McDermott is on the team, and I've been working closely. She's been keeping me organized. That's Emma Pratchett. Thanks again for all of your work putting today together. I'd also like to thank our sponsor for the event. That's Canadian Centre for Behavioral Neuroscience. Very generous support to make this happen. And of course, the other partners, which is the Hotchkiss Brain Institute, the Neuroscience and Mental Health Institute, and the Neuroscience and Stroke Program Integration Network. Thanks again to all of the attendees. Thank you for asking your questions. Your support for Alberta neuroscience means the world to everybody in this room, and from myself personally, again, when I'm wearing all the hats, which is being a philanthropist for neuroscience research, a venture capitalist for neuroscience innovation beyond the lab, and of course, as a patient who's navigating this world with multiple sclerosis. It is very inspiring to know that people are working so hard to make this a world, a better place for people suffering from neurological disorders. Thanks again. I hope to see all of you in person in the near future. If you have any questions for myself personally, feel free to reach out. The team at Campus Alberta Neuroscience does have my contact information. Have a great rest of your day.